Hello. Today we are going to be talking about cell communication and cell signaling. And this takes a jump to section 5.6 in your textbook. So if you look back at the rest of chapter 5, it's all about the cell and the cell membrane and how things are transported in and out of the cell. But this section is specifically talking about how cells communicate with each other. And this is very, very important because for multicellular organisms, this cell-to-cell communication is what allows the different cells in the body to talk with each other. But for unicellular organisms, they also need to be able to communicate. So when we think about cell communication, there is actually two different types. Local signaling, which is communication between cells that are close to each other, and then long distance signaling, which we're going to spend most of today's lesson talking about. But first, we'll talk a little bit about local signaling. Both eukaryotic cells, animal and plant cells, have different junctions that can communicate by directly contacting other cells, and these are called gap junctions in animal cells, and then in plant cells they're called plasmodesmata. So these are basically little spaces where signaling substances dissolve in the cytosol of one cell and they can pass freely to the cytosol of another. And this is one type of local signaling. There are some other types of local signaling where messenger molecules are secreted by a signaling cell and they can travel very short distances. So one example of these are the growth factors, which are going to be important because we'll see how growth factors are responsible for telling cells when to grow and or divide. And this is one example of what's known as paracrine signaling. So you can see with paracrine signaling, these signaling molecules get secreted by the vesicles, and they can travel a short distance to the target cells to tell them what to do next. Another type of local signaling happens with the nervous system, and this, known as synaptic signaling, basically consists of an electrical signal that moves along a nerve cell, and that electrical signal signifies the release of neurotransmitters. And these neurotransmitter molecules will diffuse in between the nerve cell and its target cell and then cause the target cell to have a response. So this synaptic signaling looks pretty similar to paracrine signaling, but you can see these are specific nerve cell neurotransmitter molecules that are diffused from the nerve cell and hit the target cell, thereby creating a response. But the most important signaling we're going to talk about in this section has to do with long-distance signaling. And for long-distance signaling, plants and animals use a chemical known as a hormone, which most likely you've heard of. In animals, the body system that regulates hormones is known as the endocrine system. So this is often referred to as endocrine signaling. And essentially, these cells are released these chemicals, the hormones, are released into the circulatory system and can travel to the target cells. So they travel through the bloodstream from one cell all the way very, very far away in the body to maybe another cell. So maybe, for example, you've got some cells up here, they're going to send a message all the way to some cells down here, and that can happen using hormones, long-distance signaling. There was a scientist named Earl W. Sutherland, and he was mostly responsible for studying how epinephrine is a specific hormone, how that hormone acts on cells. And epinephrine is one of the hormones responsible for regulating our fight or flight response. So you might see that used as an example throughout this section. But he basically said that cells receive signals that undergo three processes. So first is reception, then transduction, and then response. So we're going to talk about each of those in the lesson today. When we start with reception, essentially a target cell detects a signaling molecule, and typically that signaling molecule is going to bind with the receptor molecule. Then in transduction, typically we have at least one step, but usually there's many steps that convert the signal to a form that brings about our response. So the molecule usually will change shape during transduction, or it might send messages to other molecules that will change shape. But it's usually a series of events, so typically this is known as a signal transduction pathway. And then eventually we get the response, which can be any variety of activities in the cells that result from these hormone receptor molecules receiving the signal from the hormone. So first we're going to talk about reception. And this section is when the signaling molecule binds to a receptor protein. So only certain cells can receive the signals from hormones, and this is all predetermined by the type of cell. So that explains why certain hormones only affect certain cells, and the cells that are affected are often referred to as the target cells. So the receptor protein on the target cell allows for a signal detection, and then it can activate the beginning of the response. The signal molecule and the receptor come together kind of like a lock and key, and this binding is known as a ligand. So the signal molecule acts as a ligand, which essentially means it allows for the binding to the receptor to happen. And like I said, this is very specific. Like a locking key between the signal molecule and the receptor, and that changes the shape of the receptor. Once the ligand binding occurs, the receptor changes shape, and that usually signals the activation of the receptor. So this is another example of allosteric regulation, where the shape of a molecule affects its function. So we have two different types of receptor molecules. Like it says here, most are plasma membrane receptors. The other type are intracellular receptors. But we're going to start by talking about receptors in the plasma membrane. So these are receptors that are located on the cell's surface, and many are transmembrane proteins, meaning they span the whole membrane. If you remember, they go through the hydrophilic and the hydrophobic layer. And these are water-soluble signal molecules that can bind to specific sites on receptor proteins that span the plasma membrane. So these are our plasma membrane receptors. And there are two major types of plasma membrane receptors. So we have G protein coupled receptors and ligand-gated ion channels. The G protein coupled receptors, known as GCPRs, are a cell surface membrane receptor. So these are plasma membrane receptors, and they work with the G protein. So the G protein essentially binds GTP, and binds to GTP, which is, if you recall, kind of a similar molecule to ATP, just used for different functions. And then the GTP molecule then will bind to an enzyme. And there are many, many different types of G protein and GPCR pathways that cause a lot of different responses. They have very diverse functions. So here you can see a little bit of what this looks like. It's loading. So here we have our signaling molecule coming in, and we have our GPCR, the G protein coupled receptors. It receives the signal from the signaling molecule. That ligand binding causes the G protein to be activated, which will then bind to GTP. And then typically what will happen is, now that this G protein is activated, it can go and activate this enzyme and then produce some response in the cells. The other type of membrane receptor is known as an ion channel receptor, or ligand-gated ion channel, and it essentially acts as a gate or passageway for ions when the receptor changes its shape. So this is pretty common to act as a gate to open or close for different ions. The most common ions that use these channels are the sodium Na+ ion and the Ca2+ ion. And these you've probably heard about before because they are very important in the nervous system. But if you remember, ions cannot pass through the membrane on their own. They need the help of both a protein and that energy from the ATP. So here's our ligand-gated ion channel. You can see there's kind of a little space here, but right now the gate is closed. We've got some ions outside of the cell, and then here's inside of the cell. The signaling molecule acts as a ligand by binding to the gate. The gate opens and the ions are able to diffuse through into the cell and then produce a cellular response. Once the response has been taken care of, the signal receptor molecule and the ligand signaling molecule detach, and the gate will close and ions will no longer pass through. So that is one example. Then we have what's known as intracellular receptors, and these are receptors located inside of the cell. So they're either found in the cytosol or the nucleus of the target cells. Small polar and nonpolar ligands can diffuse through the membrane and enter the cell and bind to those receptors to activate them. But typically these tend to be hydrophobic chemical messengers. And a lot of steroid hormones are good examples of intracellular receptors. So those steroid hormones have those large hydrophobic regions, and they can diffuse through the membrane and activate receptors on their own. And then in both plants and animals, nitric oxide, or NO, can diffuse and create a cellular response as well. So we're actually going to stop here and pick up with the other two steps, transduction and response, in another video.